Why don't we get started? Hello and welcome. My name is Mitch Sanders. I'm here with Kyle Quinn. And we have a very special guest, Allison Ramey-Ward from Progena Care. What's the new company? Progena Global.
Progena Care Global.
Thank you, Progena Care Global. And it's a wonderful paper, and I'll let Carl start with the questions, and we'll go from there.
Sure. So let's talk about some of the just kind of intro concepts here. And keratin's quite abundant. We know it's present in epithelial linings, and of course hair, fur, and feathers. Could you tell us how you sourced your keratin and how you created these keratin matrices?
Yeah, so our keratin is actually sourced from human hair. We literally collect hair clippings, and then we use a process called it's based on a process called the Shindai method, which is a reductive extraction. So it breaks down the disulfide bonds that are so abundant in keratin and give it that mechanical strength that we know it for. So it kind of breaks those down and puts it into sort of a liquid form that we then cast into our matrix and allow it to basically recross-link to create the solid products.
And this is all from barber shops, right?
Yep.
Pretty cool. Yeah, and obviously there's a lot of hair available through through that method. And when I think about human hair, I think about how that varies person to person. And we know that melanin content then varies person to person, dictating the color. Does the keratin content in the hair vary person to person, or do you have to worry about that sort of donor to donor variability?
So we haven't observed anything like that. We do see differences in the color, but a lot of times the keratin that we extract is from, multiple different people, go to these salons and donate their hair. So we get a lot of variety in each lot of hair that we convert into the extracted keratin. So as far as our validations have shown, it's all pretty much the same by the end.
Yeah, probably just averages out with enough enough clippings from enough people getting their hair cut.
Can I ask one technical question? So the keratin has, most of it is the keratins, right? And then there's about 6% total that is keratin associated proteins, and then you had a spurious calmodulin like protein that was there, right? So the question I have is that, were those percentages by weight or were they molar? I think they're by weight, but I don't know. The reason why I ask is on a molar basis, the caps may be much higher, but on a because they're very small molecules compared to the keratins, we're very big proteins.
Yeah, so the percentages reporting is the percentage of the total proteins that were
right. So it's probably picogram or something like, or yeah, picogram per sample or something like that. Mm-hmm. And so yeah, it's not on a molar basis. The caps are actually almost probably as high as the keratins. That was my one point. Neat. Could you talk about the findings from the study? It was a really well designed, well written study.
Thank you. Yeah, so this is a combination of some work that was done before I started, but I kind of went into, with my background in biomaterials, I went into really characterizing the material itself. So we start with a little bit of basic, hydrogel material science, looking at the stiffness of the material, the water content, that kind of thing. We did a lot of proteomics, as you alluded to show, what it's actually made of. And we do see that it's made almost entirely of keratin. I mean, it is a hydrogel, so the product itself is almost entirely water, but the parts that aren't water are almost entirely keratin. There were a few, as you mentioned, other kind of proteins that we got hits for in that proteomics. You mentioned the calmodulin-like proteins, and those are things that we definitely want to keep digging into. We noticed that the product is pretty robust, this human keratin matrix. It's fairly stiff. It's almost 1 megapascal as its ultimate tensile strength. So that was pretty interesting, especially for hydrogel. Those are typically much softer. And we see that it's fairly resistant to hydrolysis as well. So again, that was something that we weren't necessarily expecting, but was very interesting. we looked out to 7 days for that. And
Can
then
I-
we kind of, oh yeah.
Go ahead. I'm waiting for you to talk about the animal study.
Okay. Yeah, we did a good bit of cellular work in vitro, looking at how human epidermal keratinocytes and dermal fibroblasts actually interacted with the keratin matrix and how that modulated their cytokine expression. And then, yeah, we eventually went into looking at, how well this product can support wound closure, and then also comparing it to some other kind of similar biomaterial-based products. So we used the DBDB diabetic mouse model. We created multiple wounds on the dorsum of the mouse and then treated it with either the keratin products, we had a human amniotic membrane product that we tested, a bovine dermal collagen, and then a porcine decellularized small intestinal submucosa products. So looking at these different sort of matrix-based products and seeing what the differences were. And we did find that keratin really helps support that wound closure as well, or in some cases even faster than some of these other matrix-based products.
What was the age of the mice?
They were, I believe it was 12 weeks at the start of the.
Okay. So we do 9-week-old mice typically, and our wound closures are much faster. Usually 24 days. The control's almost fully healed, and these were much more delayed. Do you think it was because the age of the mice, or do you think the iodine may have had a kind of an overall slowing effect of on all the wounds?
Probably a combination, yeah. So in our procedure, we used cadexamer iodine as like kind of a slow release antimicrobial, just because the DBDB mice, they can be a little more susceptible to infection. So that could definitely be part of it, and the age could contribute as well.
No, it was pretty interesting to see some of those human amniotic chorion-like materials be very slow. I've noticed before that, they do okay in the proliferative phase, but they do nothing for the inflammatory phase. And I'm guessing that the keratins just abolished the inflammatory phase. And what we saw when we did a DB study for Progenic Care Global way back when, we saw that the inflammatory phase was almost non-existent. So typically the mouse, when you take these wounds and start them, day 3, 4, they're really hot. They're actually 20% larger. And when you use this product, it seemed to completely abolish the inflammatory phase. Is that what you saw in the mice, or?
Yeah, that's definitely one thing we noticed. If you look at the healing curves in that figure, is that the biggest difference between the products is seen much earlier on. And we do see that kind of early spike where the wound size increases within that first week and then starts to go down. But yeah, we definitely see what would suggest a different sort of approach to wound healing for the keratin, because we do see that difference in those earlier weeks.
And we just to let you know, we have a 300 probe code set for nanostring. So if you ever want to look at all the phases of healing, macrophage plasticity, fibrosis and scarring, it's a great code set. It even covers oxygen and nitrous oxide responsive genes. So it's a great panel. And then what I love about doing nanostring, it doesn't have the amplification bias that PCR can have. And also there's very little pipetting steps, so there's almost no pipetting error. And the last thing is you don't have to do the analysis. It's all done for you in Roslyn bioinformatics. And it's beautiful to go from purified RNA to a day later, you've done all the nanostring, and one day later you have the data for publication. It's just amazing.
That sounds great.
And then the last thing is that the cool thing about nanostring, because it's probe based, is that it has no variance from the RNA sample. So if I took RNA and I sent it to 5 different labs, we'd all get the same results, which is pretty compelling and fun. So keep it in mind. It'd be fun to do a broader study to look at some of these biomarkers. I don't know if that PCR was done a little while ago, but it seemed like you already knew which markers to look for because you didn't look at a lot of markers.
Yeah, so for the PCR, we looked at factors that are associated with keratinocyte activation. So there's a really great paper called Keratins and the Keratinocyte Activation Cycle. It was published a little while ago, but it's a really good review of how keratins interact with the keratinocytes and kind of the wound healing cascade. So yeah, that's where we got the the IL-3 and the EGF for that PCR that we did. But then we wanted to dig in a little deeper, and we went into that microarray, which was protein-based. And this is a human cytokine microarray. It's provided by or done by a company here in the Atlanta area.
Ray Biotech, we know them really well. Yeah. So was that the quantibody array? Was it 80 cytokines, or how many did you do? If you don't remember, we use the quantibody arrays so often that I don't remember which. It really depends on the panel.
I should remember.
It's okay. That's fine. But it's a really good list of cytokines. Does it also have growth factors in that one, or no?
It does have some growth factors, yeah. So it looks at EGF, TGF beta, IGF-1,
cool.
Some basic growth factors there.
So what do you think the next steps are for the project? Where are you going? What do you think the next big thing should be?
Well, we're definitely looking at the interaction of keratins with the immune system. So one of the things that was kind of interesting, you mentioned that keratins are generally anti-inflammatory, and those keratin-associated proteins as well. But if you look in our microarray, we actually see an upregulation of proteins associated with acute inflammation and neutrophil recruitment, which at the surface level, that doesn't seem like it would be a good thing. But as we're learning more as a field about the role of neutrophils in wound healing, they actually act to sort of regulate the rest of the inflammatory process. So we'd like to dig into that a little more. We also only looked at 2 cell types here, the keratinocytes and the fibroblasts, but of course there's a plethora of immune cells that are involved, and vascular endothelial cells, kind of looking at angiogenesis potentially.
Great.
There's a lot of different ways that we could go with this.
So this product's it got approval, what, 2 years ago? I'm trying to remember when it got approved or cleared.
At the very end of 2019.
That's right. And the is this part of the products that have an issue with the MACs right now? Is this in the low bucket of the MACs, or how is this product priced and reimbursed? I should have had Howard on the call.
You're asking R&D about this. I know that we have an ASP per square centimeter. We have a Q code reimbursement, and I believe we are high bucket for the hospital reimbursement.
Okay, and is this one of the? Was this kind of singled out from the MAC's point of view, the LCDs? Right now is something that's not covered until you have clinical data, or no?
I don't believe we're currently on the nice list, quote unquote.
Okay.
But
I think there's going to be a lot of pushback. I think a lot of these products are going to need some RCTs. They may not need to be exhaustive, but they need to have some good clinical evidence because of the cost.
Definitely,
associated with it.
And we, if I can tease a new paper that we're going to have coming out soon. We recently did some clinical work with Dr. David Armstrong looking at our product in DFUs. So that should be published here in the next couple months.
Wonderful. And you had a really cool Gen 2. Is that going to come out, or is that going to be submitted, or what's the status on that?
We do have a new product in the works that we're hoping can kind of iterate on Progenimatrix and potentially look at, some different different applications. Right now, our biggest size for Progena matrix is a 10 by 10 square centimeters. So we're looking at something that we can make a little bigger and maybe be able to target, some of those larger chronic wounds or even something like burn and trauma.
Yeah, I was thinking, you don't have indications yet for burns, do you?
We are indicated for 1st and 2nd degree burns.
Oh, great. Kyle, you got to see this product. It almost feels like a tough fruit roll-up. Kind of, and the original Gen 1's a little brittle, but it works really well, but you have to be a little careful how you handle it. But Gen 2 was more flexible and a little thinner and maybe better for acute wounds or other, surgical site. Have you ever looked at scarring to see if this reduces scarring?
Not in any official capacity. Anecdotally, some of our users have reported that the wounds look better than what they would be used to seeing, but we don't have any real data associated with that.
Well, I think it was just wonderful. And you say the next paper is coming out soon. Is it going to be in WRR or do you know which journal already?
I don't know. Dr. Armstrong and Dr. Zellen handled all of that. So I know it's been submitted. I'm not sure where.
Wonderful. Anyway, Kyle, did you have any more questions?
I think we probably
wonderful. Covered the gamut. Allison, the work is spectacular. Thank you for submitting your paper to WRR. And if you ever want to be on a committee, Kyle would like to introduce you to the membership committee, because Kyle's now the chair of the membership committee. I'm just the supporter of all the committees now, which is fun. But we'd love to have you get involved with the Wound Healing Society. It's really a blast.
Yeah, absolutely. Awesome. We always need people in communications and membership. So
it's the biggest committee because we do we do a lot of the heavy lifting. Without PRRI, we wouldn't be able to do anything. But it's a really strong committee that gets to do a lot of things and gets to collaborate with all the other committees. So.
And you can do fireside chats.
Yes. Anyway, thank you for the wonderful papers. Great to see you and look forward to seeing you at the next meeting.
Thank you so much.
Thanks. Have a wonderful day. Cheers.
You too.